Greetings everyone, I am Shreya Dayanand, a Microbiology Honours student at the University of KwaZulu Natal in Durban, South Africa. My research will be focusing on determining the prevalence of pathogenic protozoan and helminth parasites in treated and untreated wastewater samples from wastewater treatment plants in the Durban Metropolitan areas. So my research was completed under the supervision and co-supervision of Dr. Maningi and Dr. Mulantwa respectively, with the additional assistance from Professor Adeleke. The South African Water Services Act 108 of 1997 states that everyone has a right of access to basic water supply and basic sanitation. Wastewater treatment is a critical process that ensures safe water for, in some cases, human consumption and the protection of the environment from pollutants. However, despite advances in wastewater treatment technology and engineering, pathogenic parasites such as protozoans and helminths still pose a significant threat to public health in South Africa. The parasitic presence in wastewater effluent that is discharged into rivers can infect communities that interact with the river water. These parasites can cause a range of infections, including symptoms such as dysentery and diarrhea, particularly in individuals such as children and immunocompromised. The current study will aim in informing the public health about the need for interventions aimed at reducing the health burden imposed by waterborne parasitic infections in South Africa. By uncovering and understanding the prevalence and distribution of pathogenic parasites in wastewater, more efficient surveillance methods can be utilized to prevent the spread of infections and curb infection outbreaks. Considering the high HIV infection rates, which are currently estimated to be approximately 8.2 million in South Africa, pathogenic parasites can cause opportunistic infections in these individuals with compromised immune systems and worsen the condition. As parasites develop resistance to water treatment methods, it is important to monitor their prevalence in wastewater, considering that many informal dwellings, such as rural areas in southern Africa, rely on water as a primary water source. Pathogenic parasites take advantage and spread rapidly within these communities. Therefore, monitoring the parasites in wastewater can aid in controlling these infection rates and optimizing treatments for resistant parasites. So by exploring pathogenic parasites in wastewater, more effective wastewater treatment technologies can be developed and implemented. Specifically, evaluating the ability of parasites to resist treatment with chlorine can reveal health hazards posed by pathogens which threaten the standard of public health provision due to their persistence in chlorinated bodies of water such as public pools, leading to an increased risk of waterborne parasitic infections. This causes a significant threat to public health maintenance, as the spread of these waterborne parasitic infections would be difficult to track. Moving on to helminths and protozoans specifically, protozoa are single-celled organisms that can reproduce both sexually and asexually. Intestinal protozoa are excreted with feces of an infected individual and exist as cysts or trophozoites. In the sewage sludge in wastewater, only the cysts can survive. And when a host comes into contact with the wastewater, the cysts begin the infection cycle by releasing trophozoites in the small intestine, which cause the infection symptoms such as dysentery. Pathogenic protozoa have shown significant negative impacts on public health. This observation is particularly significant in developing countries where some parts of the population have little to no access to adequate sanitation and potable water services. Cyclosporiasis, caused by Cyclospora species, is an infection that is evident in developing countries through contaminated food and water as a result of lack of proper hygiene facilities. It also affects developed countries through consuming contaminated foods. The surveillance of this parasite and its associated disease in the United States between 2011 and 2015 reported 10 outbreaks within this period, of which 5 outbreaks were confirmed to be from contaminated food, some of which were imported. This indicates the need for efficient parasite screening methods in both developed and developing countries alike. Microsporidia species involved in co-infections can also be threats to public health, as the parasitic infection would exacerbate the symptoms of the infection. The co-infection incidence between Microsporidia and Mycobacterium tuberculosis was seen to have a correlation. Since in a specific study, 39.6% of patients that tested positive for TB were also infected with Microsporidia species. Cryptosporidium co-infection with HIV can also occur and worsen the symptoms experienced. In a study done in Ethiopia, it was shown that the prevalence of Cryptosporidium infection in persons living with HIV was 11%, where people living with HIV with a lower CD4 T cell count of less than 200 cells per cubed millimeter had a higher chance of contracting Cryptosporidium infections. Similarly, another study also reported that a lower CD4 T cell count, which is experienced by people living with HIV and AIDS, makes the host susceptible to Cryptosporidium infections, which further impacts the immunity of the host. So this allows for the symptoms to be exacerbated, particularly within persistent diarrhea seen in co-infections with HIV and Cryptosporidium. Considering that most communities in developing countries that are exposed to contaminated bodies of water overlap with communities that do not have access to reliable healthcare, the incidence of people living with HIV and AIDS exposure to Cryptosporidium through contaminated food or water within these communities is high. Moving on to helminths, which fall under another class of parasites and are ubiquitous in various environments. These multicellular nematodes also cause opportunistic infections in immunocompromised individuals. Their life cycle involves 2 or more hosts, as the helminth's eggs are released into the environment through the feces of an infected host. The eggs then hatch and become larvae, which infect new hosts by invading different tissues and organs, where they develop into adult worms and carry out their life cycle again. Soil-transmitted helminths are a group of helminths that are present in varying concentrations in wastewater and cause symptoms such as vomiting, diarrhea, and malnutrition when infected with these parasites. The prevalence of helminths increases in developing countries due to lack of access to proper sanitation services. So the efficacy of wastewater treatment does not always have an effect on helminth removal, as helminths, unlike protozoa, can be in the form of eggs, larvae, or adult worms. Therefore, in this way, many helminth species escape treatment processes of wastewater treatment plants and are present in the effluent, which becomes exposed to communities that use this contaminated water. Similar to protozoa parasites, helminths can also cause significant threat to public health through their origins in many contaminated sources. Specifically, Nectar americanus is a type of hookworm helminth parasite that has had many implications on public health. A correlation was observed between N. americanus infections and people living with HIV and AIDS, as they exhibit lower CD4 T cell counts than those living with HIV only, due to the parasitic infection. And because of this co-infection, the parasitic and viral infection puts the individual at an immunological disadvantage. When this interaction was tested on mice, it was shown that N. americanus infection actually affected the ability of the mice to control the viral infection. The lack of a wide range of anti-helmintic treatments emphasizes the need for more clinical studies to be done on treatments available for helminth infections. Not all communities that are at risk of being exposed to contaminated water or food interacting with animals that have helminth infections also have access to reliable healthcare. However, the establishment of broad-spectrum anti-helmintics offer a possible solution if implemented in these areas as preventative methods through processes such as deworming. The aims and objectives of the research done was to determine the prevalence of pathogenic parasites in treated effluent wastewater samples. So this was done through the modified Berliner method, mini flow tag technique, and PCR. And to analyze the physical-chemical properties of the wastewater, an HQ probe and mercury thermometer was used. To evaluate the prevalence of resistant parasites that are resistant to chlorination, we performed these methods on the post-chlorination samples. This is a key as to how the samples have been coded and how I will refer to them within the contents of this presentation. So there were 2 wastewater treatment plants that were sampled from, hence A refers to one plant and IS refers to another. I and E refers to influent and effluent respectively, where H and PC refer to humus tank and post-chlorination samples respectively. It should be noted that plant A did not perform chlorine dosing, therefore there was no post-chlorination samples from this plant, and the effluent was untreated. Sampling was done using grab samples, where 3 5-liter samples were taken from influent and effluent sampling points at plant A, respectively, and 1 5-liter of influent, 2 5 liters of humus tank, and 3 5 liters of post-chlorination samples were taken from the second plant IS. So the different physicochemical properties of the wastewater in which the protozoan and helminth cysts and eggs exist impact their survival. Some temperatures could promote the death of the cysts and eggs, while others could induce a dormancy stage. These parameters are important to analyze in order to understand why the resultant prevalences came to be. So the temperature of the samples were measured with a mercury thermometer immediately after sample collection, and the rest of the physicochemical readings were done using the HQ probe. Detection of helminth and protozoan eggs and cysts can be performed using mini-flow-tac technique, which uses the principles of centrifugation, flotation, and density to obtain helminth and protozoan egg and cyst counts in a sample. This allows for the identification of cysts and eggs, which makes it useful in detecting the presence of both protozoans and helminths using one assay. The mini-flow-tac technique has a higher sensitivity compared to the flow-tac technique, and the use of zinc sulfate solution further increases sensitivity compared to a sucrose solution, and provides increased detection of parasites compared to the Cato-CATS and standard McMaster methods. Similarly, the modified Berlinger method also allows for the detection of both protozoan cysts and helminth eggs in a sample, and results in the quantification of the number of eggs present by generating the number of eggs per gram. This method uses a flotation solution and various washing steps with zinc sulfate and acetoacetic buffer to separate the sample into various layers by density differences, of which one layer would contain the protozoan cysts and helminth eggs. Using both the modified Berlinger method and mini-flow-tac technique increases the reliability of the results obtained and allows for comparison to be made between the efficacy of parasite detection of both methods. Molecular screening methods used, and this includes PCR. So this involved DNA isolation using the Quick DNA Fecal Soil Microbe Mini Prep Kit by Zymer Research. 18S rRNA primers were used for the detection of protozoan and helminths. A nested PCR approach was also applied for the detection of protozoan species, which involves the usage of 2 sets of primers, one set to anneal within the bigger range of the target sequence and slightly outside of its range, and the second set using the product from the first round will anneal to the target sequence more specifically. In summary, the modified Berlinger method, along with the mini-flow-tac technique and PCR, is used in the current study to identify the prevalence of pathogenic parasites in wastewater effluent. This table represents the physicochemical parameters for the wastewater samples collected. These were measured within 24 hours of sample collection, except temperature, which was measured immediately after sample collection for accuracy. One sample from each of the sample categories, influent, effluent, humus tank, and post-chlorination, were analyzed. A general temperature decrease from influent to effluent was seen in each plant, while all samples remained approximately neutral. The total dissolved solids increased from influent to effluent in plant A, but decreased from influent to effluent in plant IS. There is a directly proportional relationship observed between the conductivity and salinity for the samples taken, as the higher conductivity also suggests a high salinity. As seen in this table, the temperature fell within the range of 20 degrees to 26 degrees, which is below the temperature reported to cause parasite egg and cyst inactivation. This physicochemical property is supported by the pH being neutral, which allowed for the persistence of helminth eggs and protozoan cysts, due to them generally being inactivated at pH values exceeding 11.9. Therefore, the neutral pH established across all samples also encourages an optimal environment for the persistence of parasite eggs and cysts. Considering that the physicochemical parameters of the water did not inactivate the helminth eggs and protozoan cysts, the detection of parasites in the samples can be attributed to this as well. This table displays the decrease in average EPG from influent to effluent, both in plant A and plant IS, using the mini flow tag technique. This is unexpected as plant A did not perform chlorination. Therefore, the effluent was left untreated and should not have a lower EPG than the influent, ideally. A possible reason for this would be the death of the parasites that were present in the influent due to the intolerance of the physicochemical parameters of the wastewater. Hence, they were not represented in the effluent. The decrease in EPG counts in plant IS from influent to humus tank to effluent correlates with the normal wastewater treatment procedures and proves the removal efficacy of the treatments. However, there were parasites detected in the post-chlorinated samples. This would be possible through the persistence of protozoan and helminths that can tolerate chlorine dosage and therefore remain in the treated wastewater effluent. Despite parasites still being present in the post-chlorinated samples, the impact of chlorine dosage on parasite survival in wastewater can be justified as the plant A displayed higher average EPG counts, which were 58, for the untreated effluent compared to the post-chlorinated samples from plant IS, which displayed an average of 8 EPG. This significant difference is owed to the chlorine dosage as a better treatment option compared to the untreated effluent. The mini flow tag technique also allowed for the detection of helminth morphology via microscopic analysis, shown in the figure in the A. This suggests a higher parasitic prevalence than obtained using PCR, as only protozoan parasites were detected using PCR. The morphology resembles that of Trichoris species, which is a prevalent helminth parasite often detected in wastewater treatment systems. The comparison of the morphology observed can be compared to that in literature, as seen in B in this figure. This table portrays the EPG counts achieved by using the modified Berlanga method. The overall trend in this table resembles that of the previous one, where the average EPG count decreases from influent to effluent of each wastewater treatment plant. Similarly, this is an unexpected for plant A, as there is no chlorine dosage. However, the average EPG for influent and effluent of plant A in this table showed to be less than obtained in the previous table. So the difference between the average EPG of the influent and effluent is smaller in this table compared to the previous one, suggesting the superiority of the mini flow tag technique in microscopic analysis of parasites in wastewater samples. This table displayed an average EPG count of 0 for the post-chlorinated sample, suggesting that according to this method, there was absence of chlorine-resistant parasites. The successful application suggests the presence of Microsporidia species in the wastewater samples. As displayed in this figure, the band observed in lane 2 as the negative control could be a result of non-specific binding or contamination present in the sample. That's why when repeated, there was no amplification seen for the negative control in the second gel, as seen in the figure, while the positive control is still present, suggesting the reliability of this PCR process. As shown in the gel images, the prevalence of Microsporidia species within the sample using PCR is that 11 of the 12 samples analyzed displayed successful amplification of the target gene for detection of Microsporidia species, as the amplicon size obtained matched that of the expected size range, being between 410 to 420 base pairs. So looking at the presence of Cyclospora caetanensis detected in the wastewater samples using PCR, it can be seen that 7 out of the 12 wastewater samples tested positive for having presence of Cyclospora caetanensis, as there was successful amplification of the target gene for the detection of the species. This suggests that certain species of Cyclospora caetanensis are existent in chlorine-resistant strains within this wastewater system, as the post-chlorinated samples also had a successful amplification of the target genes for the detection of the species, as the amplicon size that was obtained was the same as the expected amplicon size, that being 290 base pairs. Cryptosporidium species only showed a 25% prevalence in the wastewater samples, as 3 out of the 12 samples showed successful amplification of the target gene, where the achieved amplicon size correlates with the expected amplicon size of 171 to 183 base pairs. The graph on the left shows the prevalence of parasites in the influent and effluent of each plant, and it indicates that the parasitic prevalence of the effluent in plant A is less than that of plant IS, despite plant A not performing chlorine dosage. However, this could be simply due to the parasitic prevalence of the influent in plant A being less than plant IS in the first place. This can be supported by the overall parasitic prevalence in each plant, as plant IS was 67% and plant A was 50%. The different treatment systems allowed for the development of different parasitic prevalencies, which is also displayed in a similar study where the parasitic removal rates differ between the different wastewater treatment plants. However, all plants exhibited a decrease in parasitic prevalence from influent to effluent, much like the current results on the screen. So the graph on the right depicts the difference in parasitic prevalence between the 3 protozoan species detected using PCR. These species were Microsporidia, Cryptosporidium, and Cyclosporus species. And it can be said that the Microsporidia species is predominant since it occurs at the highest prevalence, that being 92%. Hence, the conclusion of it being predominant in the wastewater treatment plants, as Cyclospora cayetanensis shows a prevalence of 58% and Cryptosporidium species is lowest at 25%. The predominant presence of protozoa parasites is also expressed in a similar study, as they found protozoan parasites in 100% of their samples. The presence of Microsporidia and Cyclospora cayetanensis in the chlorinated samples suggests the chlorine resistance displayed by these parasites in the wastewater treatment systems. Therefore, it can be seen that through microscopic and molecular analysis, the prevalence of parasites in the effluent of each plant was significantly less. So there was a greater than 10% difference between influent and effluent. However, parasitic presence was still detected in chlorinated samples, indicating the presence of chlorine-resistant protozoan species, Microsporidia and Cyclospora cayetanensis specifically. The mini flow tag technique seems to have offered a better detection of parasites in the wastewater samples, as the detection of number of parasites was greater than that obtained by the modified Berliner method, deeming it a more reliable preparation method of the subsequent microscopic analysis of the wastewater samples. The presence of helminths in the effluent is suggested by the microscopic analysis displaying a parasite in an effluent sample with similar morphology to the Trichoris species of helminth species. The greater presence of parasites in the effluent of plant A compared to that of plant IS suggests lower water quality of the effluent due to lack of chlorine dosage. This is untreated effluent and would still be exposed to communities and animals that interact with the nearby river into which the effluent is being discharged. This would increase the risk of parasitic infections within these communities and animals. Limitations of the study include the absence of metagenomics and culturing of parasites due to the observation of a wide range of species. Therefore, many parasitic species went undetected due to the lack of species-specific primers for their identification. But the presence of parasites in the chlorinated effluent from IS suggests the presence of chlorine-resistant parasites and would also suggest the need for another treatment strategy, such as UV radiation, to supplement chlorination for removal of chlorine-resistant parasites. Considering that effluents from both wastewater treatment plants are discharged into nearby rivers, the parasitic prevalence in the effluent need to be better monitored and controlled, as communities and animals are put at risk when they interact with the contaminated river water, specifically with the chlorine-resistant parasite. A combination of treatment processes would be effective in producing a high removal rate of parasites and increase the overall quality of the effluent, which can be deemed safe to be exposed to vulnerable communities. Lastly, I would like to acknowledge the following for guidance and support throughout my research endeavors this year. So this brings us to the end of the presentation. I thank you for your time and attention, as well as SciTech for giving me the opportunity to present my work. Thank you.